Let us talk about the endoplasmic reticulum and its functions now. These structures are formed primarily of lipid bilayer membranes, similar to the cell membrane, and their walls are loaded with protein enzymes that catalyze the synthesis of many substances required by the cell. Most synthesis begins in the endoplasmic reticulum. The products formed there are then passed on to the Golgi apparatus, where they are further processed before being released into the cytoplasm. First, however, let us note the specific products that are synthesized in specific portions of the endoplasmic reticulum and Golgi apparatus. First, protein synthesis occurs by the rough endoplasmic reticulum. The rough endoplasmic reticulum is characterized by large numbers of ribosomes attached to the outer surfaces of the endoplasmic reticulum membrane. Protein molecules are synthesized within the structures of the ribosomes. The ribosomes extrude some of the synthesized protein molecules directly into the cytosol, but they also extrude many more through the wall of the endoplasmic reticulum to the interior of the endoplasmic vesicles and tubules into the endoplasmic matrix. Secondly, lipid synthesis occurs by the smooth endoplasmic reticulum. The endoplasmic reticulum also synthesizes lipids, especially phospholipids, and cholesterol. These lipids are rapidly incorporated into the lipid bilayer of the endoplasmic reticulum, thus causing the endoplasmic reticulum to grow more extensively. This process occurs mainly in the smooth portion of the endoplasmic reticulum. To keep the endoplasmic reticulum from growing beyond the needs of the cell, small vesicles called vesicles or transport vesicles continually break away from the smooth reticulum. Most of these vesicles then migrate rapidly to the Golgi apparatus. Some other functions of the endoplasmic reticulum, especially the smooth reticulum, includes the provision of the enzymes that control glycogen breakdown when glycogen is to be used for energy, and the provision of a vast number of enzymes that are capable of detoxifying substances, such as drugs, that might damage the cell. It achieves detoxification through processes such as coagulation, oxidation, hydrolysis, and conjugation with glucuronic acid.
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.